It's sitting in plain sight right in the snack aisle of nearly every grocery store in the country, yet almost nobody pays attention to it. You'll find it for just a few dollars a bag, quietly placed behind the trendy seeds, behind the supplements people are told are superfoods, almost as if it's an afterthought. But its position on the shelf has nothing to do with its nutritional value or biological importance. It has everything to do with something much more uncomfortable. It cannot be turned into a product empire. It cannot be patented, packaged into an expensive capsule system, or sold through a subscription model with a glossy lifestyle brand attached. It simply grows, and you eat it. What makes this even more striking is what the peer-reviewed research has been showing for years. Inside this simple food are biological effects linked to prostate cell signaling, stem cell activity in bone marrow, and even markers associated with aging at the cellular level. And yet, despite this, it rarely gets mentioned in mainstream conversations about men's health or aging. The reason is not that the data is missing. The reason is that it doesn't fit neatly into commercial systems built around branded supplements and recurring sales. What we are talking about here are raw pumpkin seeds, a simple, inexpensive food that has been studied across multiple biological pathways. And what makes them particularly interesting is not just one effect, but the convergence of several mechanisms operating at the same time. 4 distinct biological processes, all influenced by the same food, in a way that very few commonly available foods can match, especially in relation to prostate health in men over the age of 60. A large part of the healthcare system is structured around managing conditions rather than preventing them in simple, accessible ways. That means much of the research that doesn't lead to a pharmaceutical product or a clinical procedure often never reaches the average person in a practical form. This is exactly why many findings stay buried in academic journals instead of becoming part of everyday health decisions. The idea here is not complicated. It is about translating existing research into something understandable and usable, because what is being discussed is not theory alone. It is biochemistry, enzyme activity, and nutrient interactions that have been documented in published studies, but rarely connected in a way that people can apply in daily life. One of the key compounds studied in pumpkin seeds is cucurbitrin, a plant-derived molecule that has been examined for its role in enzyme activity related to hormone conversion. Researchers have observed its interaction with biological pathways involved in the conversion of testosterone into more active forms within prostate tissue. This process becomes particularly relevant as men age, because these hormonal shifts are closely tied to changes in prostate function over time. At the same time, there's a persistent misunderstanding in general health conversations about testosterone itself. Many assume that testosterone is the direct cause of prostate enlargement, but the reality is more complex. The issue is not simply testosterone levels, but how it is converted and processed within specific tissues. That conversion process becomes the real biological checkpoint, and it is here that certain dietary compounds begin to matter in a more direct way. Even in individuals who follow generally healthy lifestyles, eat well, exercise regularly, and maintain stable weight, these enzymatic processes continue to function in similar ways with age. That is because they are not fully controlled by lifestyle alone. They are influenced by specific biochemical interactions that occur at the cellular level. This is where pumpkin seeds enter the picture, not as a miracle solution, but as a concentrated source of multiple compounds that interact with these pathways simultaneously. And to understand why that matters, we need to go step by step into what is actually happening inside the body, starting with the enzyme systems involved in hormone conversion and how they influence prostate tissue over time. In the next part, we will break down those mechanisms in detail and explore why this single enzyme pathway plays such a central role in long-term prostate changes, especially in aging men. At the center of this entire process is a specific enzyme system that becomes more relevant with age. Inside prostate tissue, testosterone does not act alone. It is converted by an enzyme called 5-alpha reductase into a more active form known as dihydrotestosterone, or DHT. This conversion is not a minor side reaction in the body. It is a central biological step that determines how strongly prostate cells respond to hormonal signals. DHT is significantly more potent than testosterone when it comes to binding to androgen receptors in prostate cells. Once it binds, it does not just trigger a small response, it activates a much stronger and more sustained signal that influences gene expression, cell growth, and tissue expansion. In simple terms, it pushes prostate cells toward increased activity, which over long periods of time can contribute to enlargement and abnormal cellular behavior. What makes DHT particularly important is not just its strength, but its persistence in signaling. When it binds to receptors, it tends to keep those pathways active longer than testosterone would. That means even small shifts in enzyme activity can lead to noticeable biological changes over time. This is why 5-alpha reductase is considered a critical control point in prostate physiology. The key insight from research is that this enzyme does not respond in a simple way to general health habits alone. Eating well, exercising, and maintaining a healthy weight are beneficial for many systems in the body, but they do not fully regulate this specific conversion process. This is why two men with very different lifestyles can still experience similar age-related changes in prostate function. This is also where the discussion shifts from general health advice to specific biochemical modulation. Certain natural compounds have been studied for their ability to interact with 5-alpha reductase activity. Among them is cucurbitin, found in pumpkin seeds, which has been observed in research settings to influence this conversion process by reducing the rate at which testosterone is converted into DHT. The mechanism is not about blocking hormones completely. It is about modulating enzyme activity at a specific biochemical step. Think of it less as shutting a system down and more as adjusting the speed of a reaction. By slowing down this conversion, the downstream levels of DHT in prostate tissue can be affected, which in turn influences cellular signaling. This is particularly relevant because prostate tissue is highly sensitive to androgen signaling. Even moderate changes in DHT levels can alter how cells grow, divide, and respond to normal regulatory signals. Over long periods, these changes accumulate and contribute to structural and functional changes in the prostate. It is important to understand that this is not a standalone explanation for prostate health. It is one part of a broader system. Hormone conversion is only one layer. Beneath it, there are additional factors such as nutrient availability, immune function, and cellular repair processes that all interact with the same tissue environment. This is why focusing on a single variable often fails to capture the full picture. The body does not operate through isolated pathways. It operates through interconnected systems where hormones, minerals, enzymes, and cellular signals all influence one another continuously. Pumpkin seeds become relevant here because they are not acting through just one compound or one pathway. While cucurbitin interacts with enzyme activity, other nutrients within the same food begin to influence additional layers of biology. This is what makes the discussion more complex than a simple one nutrient, one effect narrative. As we move forward, the focus shifts from enzyme inhibition alone to the broader nutrient profile of pumpkin seeds and how multiple compounds within the same food begin to converge on different biological systems at the same time. Beyond enzyme activity, another major factor shaping prostate health in aging men is mineral balance, particularly zinc. This is not a minor supporting nutrient in this context. Zinc plays a central role in multiple biological systems, especially those related to immune function, cellular repair, and reproductive tissue regulation. Pumpkin seeds stand out because of their unusually high zinc content compared to other commonly consumed seeds and nuts. While many plant foods contain trace minerals, pumpkin seeds provide a noticeably higher concentration per serving, making them a practical dietary source rather than a symbolic one. What makes zinc particularly important in the context of prostate biology is its relationship to cellular regulation. Prostate tissue naturally contains high levels of zinc compared to many other tissues in the body. However, research has shown that zinc levels tend to decline in cancerous or dysfunctional prostate tissue, suggesting a strong link between zinc availability and normal prostate cellular function. Zinc is also deeply involved in immune system performance. It contributes to the activity of immune cells responsible for identifying and responding to abnormal cellular changes. This includes surveillance mechanisms that detect early signs of irregular growth patterns before they develop into more serious conditions. When zinc levels are insufficient, these immune processes can become less efficient. It is important to clarify that zinc is not acting as a direct treatment for any disease. Instead, it functions as a required cofactor in numerous enzymatic reactions throughout the body. Without adequate zinc, many of these reactions slow down or become less efficient, affecting everything from immune signaling to tissue repair. One of the key concerns in older adults is that zinc absorption and utilization can decline over time. This does not always show up as a clear deficiency in standard blood tests. Instead, it may present as a subtle reduction in functional capacity, where the body still has zinc present but cannot fully utilize it at optimal levels. This is where dietary form becomes relevant. Nutrients consumed through whole foods often behave differently in the body compared to isolated supplements. In foods like pumpkin seeds, zinc is present within a complex matrix of fats, proteins, and other minerals that can influence how it is absorbed and utilized. This distinction between isolated supplementation and food-based intake is often overlooked. A supplement may provide a labeled amount of a nutrient, but absorption depends on many factors, including digestive efficiency, stomach acid levels, and interactions with other dietary components. With age, these factors can change, which is why nutrient bioavailability becomes increasingly important. Alongside zinc, pumpkin seeds also contain magnesium, another mineral that plays a significant role in cellular function. Magnesium is involved in hundreds of enzymatic processes, including those related to energy production and muscle function. In the context of aging biology, its role becomes even more critical because it supports cellular stability and metabolic regulation. Magnesium also interacts with systems involved in DNA maintenance and cellular aging processes. One of the key areas of research focuses on telomeres, the protective caps at the ends of chromosomes that shorten with each cell division. Shortened telomeres are associated with cellular aging and reduced regenerative capacity. Magnesium acts as a necessary cofactor for enzymes involved in DNA replication and repair processes. Without adequate magnesium, these systems do not function at full efficiency, which can contribute to faster cellular aging at a biological level. This does not mean magnesium prevents aging, but it does support the systems that maintain cellular integrity over time. What makes pumpkin seeds particularly notable is that they combine both zinc and magnesium in meaningful amounts within a single food source. Instead of relying on multiple separate interventions, this creates a combined nutritional profile that interacts with several biological systems at once. As we move forward, the next layer involves another class of compounds present in pumpkin seeds that influence cellular membranes themselves, adding yet another level to how this food interacts with prostate tissue biology. While enzymes and minerals influence internal cellular processes, there is another layer of biology that often gets less attention but is just as important: the structure of the cell membrane itself. This is where a different group of compounds in pumpkin seeds becomes relevant, known as phytosterols. Phytosterols are plant-derived molecules that are structurally similar to cholesterol. Because of this similarity, they can integrate into cell membranes and subtly influence how those membranes behave. This is not a superficial effect. Cell membranes are not just protective barriers. They are active platforms where signaling processes take place. In prostate tissue, many hormonal signals depend on membrane structures that organize receptor activity. These structures, often referred to as lipid microdomains, help cluster receptors together so that signaling molecules like DHT can bind efficiently and activate downstream processes. When these membrane structures are stable and organized, signaling tends to be more efficient and more intense. Phytosterols, when present in sufficient amounts, can become incorporated into these membrane regions and influence their composition. Because they resemble cholesterol, they compete for space within the lipid structure. This can subtly alter the organization of receptor clusters and reduce the efficiency of certain signaling pathways. In practical terms, this means that even when DHT is present in the tissue and receptors are available, the structural environment required for optimal signaling may be partially disrupted. The signal does not disappear, but its efficiency can be reduced. This creates a second layer of modulation on the same hormonal pathway that was previously influenced at the enzyme level. This is important because it shows that the biological effects of pumpkin seeds are not limited to a single mechanism. Instead, they operate at multiple levels of the same system. One layer affects hormone conversion, another influences nutrient-dependent cellular function, and now this layer affects how signals are physically transmitted at the membrane level. This multi-layer interaction is rare in nutrition science. Most foods or compounds tend to influence one dominant pathway. However, pumpkin seeds contain several bioactive components that act on different stages of the same biological cascade. This is what makes them particularly interesting in the context of aging prostate physiology. It is also important to understand that these effects are not isolated or independent in practice. They interact with one another. For example, changes in hormone levels influence how cells respond to nutrient availability, while nutrient status influences how efficiently enzymes and receptors function. The body is constantly integrating all of these signals at the same time. This is why a single isolated intervention often produces limited results. If only one pathway is modified while others remain unchanged, the overall system may still continue functioning in a similar pattern. Meaningful change often requires multiple points of influence acting together within the same biological network. Pumpkin seeds, in this context, provide an interesting case because they do not rely on a single active compound. Instead, they deliver a combination of molecules that interact with hormone conversion, mineral-dependent cellular function, and membrane-level signaling architecture simultaneously. As we continue into the next section, we shift from molecular mechanisms into real-world case patterns, where these biological effects appear in practical, lived experiences and how they translate into observable changes over time in aging men. When we move from biochemical mechanisms into real-world patterns, the discussion becomes less abstract and more grounded in how these processes appear in everyday life. In clinical observations and documented case patterns, changes related to prostate function rarely come from a single intervention. Instead, they tend to reflect a combination of small physiological adjustments accumulating over time. In one frequently observed pattern, men in their late 60s or 70s begin to notice gradual changes in urinary frequency, sleep disruption, and fluctuating energy levels. These symptoms are often attributed entirely to aging or prostate enlargement alone, but in reality, multiple overlapping systems are usually involved. One of the most common assumptions is that prostate enlargement itself is the sole driver of nighttime urination. While prostate size can contribute, it is not always the only factor. Bladder muscle sensitivity, hormonal signaling, and mineral balance can all influence how often the body signals the need to urinate, especially during sleep. This is where the earlier mechanisms begin to connect in a practical sense. If hormonal signaling is influenced by enzyme activity, if immune function depends on mineral availability, and if cellular signaling is shaped by membrane structure, then small imbalances across these systems can create noticeable functional changes even without dramatic structural disease. In some documented patterns, individuals who begin incorporating nutrient-dense foods like pumpkin seeds into their routine report gradual shifts rather than sudden changes. These shifts are not uniform or guaranteed, but they often appear in sleep quality, nighttime urgency, and overall energy stability. It is important to interpret these observations carefully, as they are not controlled clinical trials, but they do reflect how multi-pathway nutrition can influence complex biological systems. One key point that often gets overlooked is the difference between treating a symptom and addressing multiple contributing factors. For example, focusing only on prostate size may miss underlying contributors such as inflammatory signaling, insulin dynamics, or mineral insufficiencies that affect bladder and prostate interaction simultaneously. This layered perspective becomes especially important when considering why some standard interventions produce partial results. If one pathway is modified while others remain active, the overall system may still behave in a similar way, even if one marker improves. This is why comprehensive approaches often involve more than one adjustment, even when the changes appear small individually. The concept here is not about replacing medical treatment, but about understanding biological complexity. The body does not isolate problems into single categories like prostate issue or nutrient issue. Instead, it integrates signals from hormones, nutrients, nervous system activity, and metabolic status all at once. Pumpkin seeds become relevant in this context, not because they are a standalone solution, but because they interact with multiple layers of this system simultaneously. This is what distinguishes them from single compound interventions that target only one pathway. Their effects are distributed across enzyme activity, mineral availability, and cellular signaling environments. As a result, any observed changes in real-world patterns should be understood as system-level adjustments rather than isolated cause and effect outcomes. This is especially important when interpreting improvements in symptoms like sleep quality or urinary frequency, which are influenced by multiple overlapping biological factors. In the next section, the focus shifts toward how these mechanisms are organized into a structured daily approach and how timing, preparation, and dietary context influence the overall effectiveness of this multi-pathway nutritional strategy.